Good morning, Class 12 Biology, Section P, Model Higher Secondary School. Ngasi noigi lessons Principles of Inheritance and Variation. Ahanbada Inheritance. Karai takilai Genetics. What is genetics? Genetics, it is the branch of biology which deals with inheritance and variation of characters from parents to offspring. Haibati branch of biology ni biology which deals with inheritance and variation and characters from parents to offspring. Haibani. Hauji, what is inheritance? Inheritance, it is the process by which characters or traits are transferred from one generation to the next. Haibati, characters say parents to offspring da generation to generation da transfer tobaki process to bu inheritance kawi. Now, what is variation? Variation, it is the degree by which progeny differs from each other and with their parents. Variation haibasina karino difference between offspring and difference between parents to offspring haibani variation haibani. Now, Mendel's laws of inheritance. Gregor Johann Mendel is called father of genetics. For the first time, Mendel conducted hybridization experiments on garden pea, Bisum sadaivum, scientific name, for 7 years, 1856 to 1863, and proposed the law of inheritance in living organisms. Gregor Johann Mendel sina ihan hanbada inheritance living organisms da kamaina character se transfer tobake haibagi mechanism of inheritance imani ihan hana study daurami. Mana study dauramisi carton P. Akoi local name da Hawaii Tharak. Scientific name da na Pisum sativum. Haiba carton P si da mana for 7 years study daurami. Now Mendel's experimental material. Mendel's na karamba material sijinaraga experiment okay habada. Mendel selected garden pea plant as a sample for experiment. Mana garden pea is magi experimental material oina select awi because of maram makhada piri singsiangu easy availability on a large scale. Many varieties are available with distinct characteristics. Lifespan of pea plant is short. They are self-pollinated and can be cross-pollinated easily. In case self-pollination does not occur. Haibati carton pea sijinabagi mana select tobagi maram samlapna carton pea se akoina yam laina phangbangami aduga large scale da phangbangami Hawaii Tharak se adui ki mathakta carton pea si ki variety makhan yam lai akoina hekta khangdokpa yaba character ko adagi magi life cycle lifespan se short oi habadi akoina study toba ganda short period short time ki manungda magi life cycle si irai laina study toba ngami aduna experiment pangthokpada yamna laiba oihalli aduga masi self-pollinated flower ni. Aduga cross-pollinated so yam laina pangthokpa yai. Maram asigidamakta mental na garden bee se choose tawbani. Then working method. Mental ki experiment maki working method si kamain tawke habada. Mental's success was also due to his meticulous planning and method of work. They are number 1 he studied only one character at a time. He used all available technique to avoid cross-pollination by undesirable pollen grains. Then he applied mathematics and statistics to analyze the results obtained by him. Mendel selected 7 contrasting characters of garden bee for his hybridization experiments. Then now contrasting characters studied by Mendel in P. Magi 7 contrasting characters in se makada piri sing sini yango sina characters in doni mana choose story ba 7 characters ko adeki sina contrasting character pair 7 pairs to sida laire siki pair se dominant and recessive matungina dominant recessive loina sida pire si characters in number 1 da stem head se tall and dwarf. Then flower colors in the violet and white ni. Then flower position dana axial and terminal. Then pod shape dana inflated versus constricted. Then pod color dana green versus yellow. Then seed shape and a round versus wrinkled. Then seed color the yellow versus green. Siki 7 pairs of contrasting characters and 7 characters say Mendel na garden P haibasi da choose tawraga si characters sing si mana study tawi. Then now next point up. Mendel conducted artificial hybridization or cross-pollination using true breeding bee lines. True breeding lines are those that undergo continuous self-pollination and show stable trait inheritance. Mendel na maki experiment si kamay tawke habada artificial hybridization ni mana experiment orisi. Haibati cross-pollinate tawba. Mana cross-pollinate tawriba artificial hybridize tawriba potse pea plants ida karamba seed sijinake habada true breeding pea lines habati tall si true breed oigadaba dwarf si true breed oigadaba. Habati true breeding lines are those that undergo continuous self-pollination habati mana breed tawnaba trait ama select tawriba plants trait se mana allowed to self-pollinate for many generations tawbakanda same trait se continuously magi inherit tawbataragati madupu true breeding lines kawe. God. Then next point, the hybridization experiments includes magi hybridization experiments haraga kari jange habaganda emasculation jangi emasculation haibati remover of anthers magi flowers idagi magi cross pollinate onabagi tamakta mana apamba character do cross pollinate onabagi tamakta anthers remove tawgadaba anther haibati male reproductive organs ni ko and transfer of pollination magi pollen se remove tawraba aduga atopa character mana apamba character sina magi flower stigma aduda pollinate tawhanba adu cross pollination ko sipuna hybridization experiment kawi. Then hojikti yengo koi inheritance of one gene. Sibu monohybrid cross su kawi ko. Haibati monohybrid cross is a cross between two individuals of species considering the inheritance of single pair of contrasting character. Example da a cross between pure tall capital TT na mosin bako and dwarf in a small TT. Haibati monohybrid cross se individual ani ki maraktani. Individual ani se tongan ba character contrasting character pair of contrasting character like ba. Example da koi tall ga dwarf se cross to we haina laurasi sibu monohybrid cross kawi. Maki monohybrid cross ta mental na kamay tawi yung mental cross tall and dwarf pea plant and collected all the seeds obtained from this cross. Then he grew all the seeds to generate plants of first hybrid generation called F1 generation. He observed that all the plants are tall. Similar observations was also found in other pair of traits. Yengu magi monohybrid cross da mendel na tall and dwarf plants da cross to baganda tall and dwarf plants si mana hanba da cross pollinator. Aduki seed si mana loina collector. Aduki seed si amuk mana thare so dore ko thare tharaba matung da haugalakba plants sing sigi appearance se kamai character si kamai toke habada ngasai ti tall ka dwarf ni mapani gi cross pollinator ka harvest of a seed to amok tharaga haugalakba plant to the only all tall plants ka haugale sigi tall plants ka haugale ipa first parents tall ka p cross to haugala ipa ahan ba tall plant ki generation sipu mental na F1 generation first filial generation have ni ko F1 have this sipu F1 generation kawi adagi mental na atopa character have the atopa 7 pairs of contrasting character na drama mana choose tawrise only tall and dwarf the mana study tawba character si atopa pairs sing da su chumbra haina mana observe tawba kanda sab mana ba result se adai da su loina phangi. Then Mendel self-pollinated the F1 plants and found that in F2 generation, some plants are also dwarf. The proportion of dwarf plants is 1/4 and tall plants of dwarf plants is 1/4 and tall plants of 3/4. Mendel na amok ngasai ki F1 plants tall ngak to tholok ba F1 plants allowed to self-pollinate. Self-pollinate haibasi F1 plant ma sense cross-pollinate nataba self-pollinate hali. Ado self-pollinate raba seed se amuk thara kanda mana haugalakba plants singse asibu mana F2 generation kaure amu. Siki F2 generation ke plants singse kamai tauke haibada ngasai ki tol ngakta oirambado haujikti F2 dadi tol su yaurei. Ado ke F1 de yautaba dwarf haibadi atien ba short plant dwarf haidi short haibani ko short plant su yaure tall ka dwarf plant animal se F2 generation da amuk phangi masi mental na calculate toraga maki proportion si yengba kanda kamai tauke habada saruk mari thopagi amati dwarf oire ko adeki saruk mari thopagi ahumna tall plants oina amuk F2 generation da mental na phangi adudagi di amuk kai daure the contrasting traits haibati tall and dwarf did not show any mixing either in F1 or in F2 generation maki contrasting trait ani se F1 generation da shu F2 da su amta mix tawte mix haibati asang ba tall sunataba dwarf sunataba marakki haibati medium size amata yaurakte tall na tall atum oi dwarf na dwarf adum oi ko then amuk yangu The parental traits are expressed in F1 generation, while at F2 generation stage, both the traits were expressed in the ratio of 3:1. Masai mental na F1 dadi only parental habati door plants na pang. Atuka F2 dadi both door ka dwarf animal si in the ratio of 3:1. The mana pangi habati 3 tall, 1 dwarf. 3 na tall oiba kanda dwarf na 1 oiba 3:1 ko si ratio si da F2 generation da phangi hojikti mental ki cross to mana ngasai ki experiment si kamai to ke habse akoi figure oina diagrammatical oina si da present or si yangu si P haibati parent ko first parent ni parent si akoi na pure tall ni cross with pure dwarf ni sigi pure tall and pure 12 से अखोइना genotype ता कमाइना present ओके हाइबदा capital TT and small tt capital TT सिना tall की alleles ni adegi sigi small tt सिना pure 12 की alleles ni ko sigi explanation next da aina point mayam amasu iri bani amuk yengsi ado hojik diagram si da ai haipetang mention tawramke Masai allele se what is allele have the next atum lakani ko allele TT sina during gamete formation da TT sigi marak sida T ama chongthore e gamete da adagi Masai dwarf ki small TT sida su gamete da di T ama khaktang yaurakani ko sida di aina amamamtang iri aduga sigi si phajana chawthongna amusu sida T ama capital sida T ama haina ani thokadabni ko aduba sida diagrammatically amamamdang ina irisi ko noina asi gyan dana pa yangu khangdraga su amuk discussion tawsi sina gamete ni si gamete ani si cross tawre haibati tol ga dwarf ani si cross pollinate tawre fertilize tawhale ko fertilization da capital masagi T ama lai small t ama capital T small t yaure siki capital T small t se sina F1 generation magi first filial generation plant siki genotypes ni magi phenotypes haibadi magi character ti kamai tawi tall oiba tall se akoina hybrid kawi hybrid haibadi magi genotype se ani si manate sumaina ani manateba sumaina manateba si ani si mix taweba different genotypes ani si different oiba asigi tamakta sigi si hybrid haibadi heterozygous haibani ko hybrid tall kawi sigi hybrid tall plant si F1 generation plant si maintain allowed to self pollinate cross pollinate na taba F1 plant masen se amuk fertilize to hale cross self pollinate to hale si selfing hai si ko si self pollinate to hale self pollinate to bakanta amuk kai tawi si ki capital T small t si gamete capital T small t chongthe eko si su capital T small t capital T small t si gamete sisu gamete ni noina sida si usi karino hana confuse lamgani sigi sign simple se male haibani female haibani ko female ni sina female gamete ni female plant ni sina male plant ni sina female gamete male gamete ko sisu akoina sina female plant ne lawsi sina male plant ne na lawsi sina male gamete ni sina female gamete ni ko gamete ani se pollinate tawdoini cross self pollinate aduna capital T capital C capital T la e magi phenotypes na tall orani si capital T small t si su tall orani si su si ani si cross over kanda capital T small t si tall ore ko lagi sigi small t si cross over da small t ani laire si dwarf plant ore ko sigi se aina sida uliba si Punnett square ni si pot se si figure se smaina square shape da orisipu Punnett square kawe ko Punnett square ni aina ulisi to understand monohybrid cross ko monohybrid cross se understand to na but kita magtani haipati yung sisi monohybrid cross understand to na ba mental ki experiment use to bani si tall and dwarf ki monohybrid cross ni to kasina F2 generation ni ko hojik point sing se makhada ai ulake point sing se sigi hojik ki cross su ki makhada yango aina sida ulisi phenotypic ratio sida na genotypic ratio phenotypes haibadi magi appearance external appearance haibadi tall dwarf tall awangba dwarf short awangba anemba akoina uriba sakse sipuna phenotype kawi magi F2 da generation da magi ratio si kaya like haibada 3 tall 1 dwarf 3:1 haibani ko percentage da 75% in a tall oi 25% in a 12 plan oi haibako and then genotypic ratio haibadi plan sida yaureba magi gene si ki genetic constitution sipu genotype kawi ko adu magi genotypic ratio se pure da tor si pure da mix da ba capital TT si sina 1 oiba ganda hybrid tor sina hybrid haibadi mix dore haibadi magi genotype ani si mana tre haiba ko mana ta ba capital T small t ani sina 2 oiba then pure dwarf small t ani si shu 1 yawi haibadi magi genotypic ratio sina 1:2:1 sina 2:1 3:1 ko simage hojiki like kan figure dugi phenotypic and genotypic ratio ni then Mendel na magi cross to habit samlapa mana explain monohybrid cross to kamai to ke habri samlapa ma ai sida point khara piri si yang si Mendel proposed that something was being passed down from parents to offspring through the gametes over successive generations. He called them factors now known as genes haibadi karigumba character ama parents to offspring then next mathang generation da sum character ama mathang pass toba gumba ama Mendel na find out tawi maduki character to bu Mendel na factors haina matam do the factors kawi hojikti akoina madubu genes haina khangnare ko then what are genes hojikti genes haibasi karino genes are the unit of inheritance they contain the information required to express a trait haibadi genes se akoi hojikti units of inheritance ni haina mayamna khangne ko mangonda kari yawi information akoi ki character mathang transfer tore iba character do genes haibasida yawi then next genes that code for a pair of contrasting traits are known as alleles akoi ngasai ki na monohybrid cross ki Punet score to noi amu ningsangu yangu aina told the capital TT na present over mi yauram gani atupu alleles kawi genes that code for a pair of contrasting traits sibu akoina alleles kawi ko genes amamam the alleles ani na mahut sili then alphabetical symbols are used to represent each gene akoina alphabetical letters se magi genes ke symbols oina sijinai capital letter haibadi TT se akoina mamangi experiment haibadi Punet score to yangu capital TT use todra aduki capital TT sina express for in F1 generation and small letters TT for other genes haibadi nangi F1 generation da capital T ama small t ama yawi ko capital T sina tall gini small t sina dwarf gini ko adu oiraga su capital ka small ani yauraga su F1 da the only tall plan oikidra adu ki tall express toriba F1 da express to bangami ba character to gi mahuti capital si use to we haba F1 generation da express to bangam hauda ba character ama yaudi yauba express ti to bangam da ba adu puna akoina small letter sijina ni haba ko when the tall TT and dwarf small TT plants and only are present and only one allele is transmitted to a gamete masai ki capital TT ka small TT si cross todra plant parents tati gene ani si yawi ko atupu gamete form to bakan tati only one allele si transmit tawgani haiba ani yaujabadu allele dadi amakhaktan yaugani haibadi the gametes of tall TT plants have the allele T and the dwarf TT plants have the allele T haibadi tall plants ki allele ani yaubasi gamete dadi only capital T yaurani adagi dwarf ki small TT yaubaduna magi gamete dadi only small t khaktan yaurani bako. This segregation of alleles is a random process and so there is a 50% chance of a gamete containing either alleles as verified by the results of crossing. Nangi crossing to ki matung ina yango masai ki parents ki TT ka sisi yaubasida ani si separate out tokiba aduga only T lakba aduga only capital T and small t lakba sibu segregation kowi sisi 50% magi cross over si 50% chance lay haibani ko after fertilization of capital TT and small tt traits hybrids contain capital T and small t and are called heterozygous to ngasai ki akhoi ki diagram tu na among yengo parents capital TT ga small tt se cross to baganda maki F1 da akhoi na phangi si capital T ga small t yaure F1 generation ki plant da di ko adana si ki plant si heterozygous kawi heterozygous haibadi hybrids or heterozygous kawi heterozygous haibadi maki genotypes ani si manati haibagini ko then Mendel found the phenotype of capital T small t to be similar as capital TT parent in appearance. He proposed that in a pair of dissimilar factor one dominates the other, that is capital T in this case, and hence is called the dominant, while the other factor small t is recessive. Ngasai ki crosso ning among yango ko haibadi F1 plants ki genotype city capital T small t ni ko parents ki na capital T animak se sika sigi animak sigi appearance magi phenotype ti tall plant oyeba appearance tadi ko animak si appearance ti tall plant oy pair ani si manata tarasu dissimilar factors isu tum tall oy ado Mendel na kari haike si ani si small short su yawi tall su yawraga kaidobagi tall oirino F1 da Mendel na kari hai sigi tall express toba ngamiba tall sipuna Mendel na dominant character kawi aduka express toba magi dwarf the express yaudi yauraga express toba ngamhodraba sigi dwarf character sipuna Mendel na kari kawi recessive character kawi ade aina ngasai so mention daukre sigi symbol ani se ko sipuna female si agoigi cross adi X na represent orani ko plant adi ki sina male ki symbol ni male gametes ki si ani si F1 da su F2 da su si symbol si loi yauram gani sina male female ki symbol ni then yango Mendel based his observation on monohybrid cross proposed to general studies called principles or laws of inheritance. Mendel na magi monohybrid cross sida based oraga mana two general rules ama obtain dawi adobo okaina principles natraga laws of inheritance haina kawi Mendel's ki laws ani tudi kari no first law da law of dominance haibasini law of dominance na kari hai law of dominance states that when two individuals of a species differing in a pair of contrasting characters or traits are crossed, the trait that appear in the F1 generation is dominant and the alternate form that remain hidden is called recessive. Then second law na law of segregation or law of purity of gametes sukaweko. This law of segregation states that the members of allelic pair that remained together in the parent segregate or separate during gamete formation, and only one of the factors enters a gamete. Students, ngasidi sida piramba aina ngasi takpa lesson si noy munna study tawro. Aduga khangdabado amuk next class da noy aiki WhatsApp group da adum thagalau. Aduda akoy amuk noy na thagalakpa madungina amuk discussion ki madam amuk lausi. Okay, thank you. See you next time.